Have you ever wondered about the different natural processes that take place in our surroundings? Even though we may not notice them, they are very important and they are occurring all of the time. In this video, we'll be covering the following themes: signal transduction, mutation, and energy flow. So let's dive in.
Before starting, let me introduce you to Emiliano Abello, our expert in the field, and the representative of Rochester School as well as the Colombian Union for Biology. Welcome, Emiliano.
Alright, so imagine a plant like a super efficient solar powered factory. Its entire life depends on sunlight, not just to grow leaves and look pretty, but to run the complicated machinery inside every single one of its cells. It all starts when sunlight hits the leaves. That light is made up of little particles called photons, and inside the leaf are special cell structures called chloroplasts. These chloroplasts are packed with pigments, mainly chlorophyll A and B, that are basically like solar panels because they absorb red and blue light best. And when photons hit these pigments, they get electrons super excited, literally. So that excitement kicks off the first part of photosynthesis, called the light-dependent reactions, which take place in the thylakoid membranes, which are like stacks of pancakes inside the chloroplasts. Here, water molecules get split in a process called photolysis, creating oxygen, protons, and electrons. Those electrons zoom down something called an electron transport chain, kind of like an energy slide, pumping protons into the thylakoid space and building up a pressure system. The protons then rush through a cool little turbine called ATP synthase, spinning it to create ATP, the plant's energy currency. Meanwhile, the electrons help maintain NADPH, another energy carrying molecule. So now, thanks to sunlight, the plant has made ATP and NADPH, basically energy stacks, ready to power other processes, and it releases fresh oxygen for us to breathe. So, I guess we may give our thanks to plants. Next comes the Calvin cycle, also known as the light independent reactions, which happen in the chloroplast stroma. This is where the plant takes carbon dioxide from the air and uses the ATP and NADPH to turn into glucose, a type of sugar. That glucose is kind of a big deal. It's used for energy, stored as starch, or turned into building material like cellulose for making cell walls. But it also powers one of the most important processes in the plant: mitosis or cell division. Now, here's where things get even more interesting and quite complicated. Mitosis is how the plant grows taller, grows new roots, repairs itself, or even reproduces. But mitosis is no joke. It takes a lot of energy, and that's when the ATP comes in. The plant uses it to build microtubules, which help pull chromosomes apart from the mitotic spindle, which is in fact the structure that moves chromosomes, and powers motor proteins that literally drag DNA around. It's like a microscopic construction site with cranes and cables and workers, all powered by ATP. Mitosis has several steps. First, the cell gets ready by growing up and copying its DNA. This occurs during interphase. That DNA copying happens in the S phase, during an enzyme called DNA polymerase. If all goes well, the cell enters mitosis. Chromosomes condense, line up in the middle, get pulled apart, and then two new nuclei form. Finally, the cell splits in two through a process called cytokinesis, like cloning itself. But here's the catch: if there's a mistake while copying the DNA, or if outside stress like UV radiation gets involved, mutations can occur.
First of all, before moving forward, it's important to recall the different effects of mutations, which, in fact, can be reduced into three effects. First of all, beneficial effects are those mutations that might help organisms live and survive, just as lactose tolerance, which helps humans break down the sugars in milk into molecules that can be used as a source of energy. Secondly, there are the harmful effects, which are damaging change to an organism, and these mutations may affect an organism's survival, such as the change in the color of the skin, which thus impacts negatively the way an organism can hide from a predator. And there are some neutral effects that are not beneficial or harmful to any organism, such as the difference in eye color in a cat. It doesn't affect its function in any way, neither its trait. Now, going back to our main idea, now picture UV rays from the sun sneaking to the leaf and messing with the plant's DNA. They can cause pyrimidine dimers, which are like little glitches where two thymines in the DNA get stuck together. This twists the DNA out of shape, like bending a zipper the wrong way, making it hard to copy properly. If repair enzymes like photolysis can't fix it in time, the cell ends up with a mutation, a permanent change in the DNA sequence. There are different kinds of mutations. A point mutation is like a typo in a single letter of DNA. Maybe it changes a word completely, called a missense mutation. It creates an early stop or doesn't change much at all. Some more dramatic are insertions or deletions, where extra letters are added or removed, scrambling the whole message, kind of like shifting every word in a sentence so it no longer makes sense. And sometimes entire chunks of chromosomes get flipped, deleted, or moved. Now, let's connect this to how the plant responds to its environment, specifically through a hormone called ABA, short for abscisic acid. Think of ABA as the plant's emergency alarm, where it's hot, dry, or stressful. ABA is released to tell the stomata, those tiny pores on leaves that open and close, to shut down and conserve water. This signal starts with ABA binding to a receptor protein in the word cells. Then it kicks off a signal cascade, including proteins like MAP kinases and ion channels like SLAC1, all working together to close the stomata. But what if a mutation breaks that system? Let's say there's a mutation in the gene that codes for the ABA receptor. The shape of the protein changes, so ABA can't bind properly. That means the stomata don't get the message to close, even if the plant is dying of thirst. Result, the plant keeps losing water, starts to wilt, and might not survive. Or maybe there is a frame-shift mutation in the gene for the SLAC1 ion channel. The channel never forms correctly, so ions can't move. The word cells don't respond, and again, stomata stay open. Or a mutation hits one of the MAP kinases, which are kind of like relay runners passing the signal down to the line. If one of them is faulty, the message stops mid run, and the stress response genes never get turned on. Before we go any further, let's talk about stomata. They're basically the plant's little mouths, and they are way more important than they look. These tiny pores, mostly found on the underside of leaves, open and close like automatic doors. Their job, letting in carbon dioxide for photosynthesis, releasing oxygen for us to breathe, and managing how much water vapor the plant let go of. Kind of like a smart thermostat for moisture. Each stoma is controlled by two word cells that puff up to open the pore when conditions are good, or shrink to shut it tight when the plant needs to save water. It's a super precise system that responds to signals
like light, humidity, CO2 levels, and even plant stress hormone like ABA. So yeah, Samara may be small, but there are total pros at environmental multitasking. Now, coming back to the main idea, what is the final effect of the different mutations? Well, the plant becomes incredibly vulnerable. If stomata can't close, the plant loses too much water, can't regulate its internal temperature, and can't take in enough carbon dioxide. This affects photosynthesis, reduces glucose production, stunts growth, and can even lead to visible signs like brown leaves, curling, or dying tissues. So, even though it all starts with something as simple and beautiful as sunlight, a single broken step from energy production to cell division to stress signaling can have huge consequences for the plant's health and survival.